Hi guys, chapter 5, going over chemical messengers. These are basically chemicals used by various organ systems to communicate with one another, uh, meaning between the organs within the same organ systems, within the same organ system, or used by a specific organ system to control all other organ systems. So the major chemical messengers are used by three major organ systems: the nervous system, the endocrine system, and the immune system. So those three organ systems rely massively on chemicals, and they release these chemicals, and these chemicals function as a messenger. So they carry a message um, onto the target cells and inducing the target cell to um, carry out a response. So the idea here is to understand the basis of these signaling cascades, from the messenger arriving at the front door, the messenger arriving at the cell, the target cell, and what happens next. And when we deal with chemical messengers, so they are for communication between cells. And when we say between cells, we are talking about intercellular. So inter means between neighboring cells, or from one cell to the next. It doesn't have to be neighbor. Oftentimes the chemical messenger travel a long distance. So it doesn't have to be short distance cellular communication. It can be short distance, it can be long distance. The point being is the communication is between two distinct cells. Those two cells can be next to each other, or those two cells can be completely within two different organ systems. Okay? And when we say communication, we are talking about signaling mechanisms. And the signaling mechanism can rely on different types of message. The message can be electrical, or the message can be chemical. So we're going to go over electrical signaling mechanisms and chemical signaling mechanisms. And in anatomy, you learn about this concept, the gap junctions. The gap junctions are 24/7 openings between neighboring cells. So from one cell to the next, there's a tunnel, there's a passageway that remains open at all times, allowing communication between the two cells, which are next to each other. We call those openings, 24/7 openings, passageways, tunnels, gap junctions. So we're gonna learn about them again, revisit the gap junctions and their importance for electrical signaling mechanisms. So those are the two types of signaling mechanisms: using an electrical signal or using a chemical signal. Once we've defined that, we're going to go over specific types of chemical messenger. Right? So when we talk about chemical messenger, we can classify them based on two ways. we can classify them based on their function. So we call it functional classification. Or we can classify them based on the way they look. So based on their structure. So structural classification or chemical classification. And then we're going to finish up the lecture with the signaling cascade themselves. So the events leading to a cell's response. So we call that signal transduction mechanism. So this is basically a cascade of events CASCADE, cascade of events, cascade of molecular signals leading to inducing a cell, a target cell, to respond to a messenger. And in this case, it can be an electrical messenger, it can be a chemical messenger. And if it's a chemical messenger, here, so I'm gonna, instead of rewrite that, I'm gonna write it here. So chemical messenger, right? CM. We need to ask ourselves, is this chemical messenger water soluble? H2O soluble? Or the chemical messenger is fat soluble? Because there are consequences to this chemical property. So fat soluble. So if it's a chemical messenger, we need to address that. Is that messenger hydrophilic, meaning water soluble, or lipophilic, meaning fat soluble? So we're going to talk about that because the consequence is it implies the location of the receptor. That's why you see here intracellular receptors or receptor at the cell surface, at the cell membrane, or at the plasma membrane. Okay. So keep that in the back burner. So this is the gist for the lecture. So let's get the ball rolling. So again, mechanisms for intercellular communication. So these are ways for cell-cell communication. So we can have what is called direct communication through a gap junction. So 24/7 openings. So this is a gap here. This is the one gap junction. This is the second gap junctions. And you're required to know these are the transmembrane protein used to build these gap junctions. They're called connexon. And these passageways remain open 24/7. So we have cell number 1 and we have cell number 2. The two cytosol are continuous with one another through these gap junctions. Okay? And this is make electrical signaling possible between the two cells. And this mechanism is important for very specific cells. For example, when we say glands, this is for glandular secretion. For example, endocrine glands that releases hormones, neurons, some neurons, and what is not on here on purpose, you need to add cardiomyocyte. Oh, it's written here, cardiac muscle fibers. Yeah. Now, let's talk about the cardiac muscle fibers first. The cardiac muscle fibers, these are cardiac muscle cells, and they are called cardiomyocytes. cardiomyocytes, cardiomyocytes. So you would have cardiomyocyte number 1. You will have cardiomyocyte number 2 sitting next to each other in a heart, and you have these gap junctions. So what it means is, if there's an action potential here, AP stands for action potential, from this cell, that action potential can spread to the next cell through the gap junctions. So this is important because what happened is you can technically activate one single cardiomyocyte in the heart. Through these gap junctions, the entire heart becomes activated through the gap junctions. So that's a powerful way to activate the heart. So when the heart is activated, it is activated as the entire organ, as one unit, right? Through these gap junctions. We have the same principle being applied to smooth muscle cells, especially those in our digestive tract, right? So these smooth muscle cells rely on the gap junctions to control smooth muscle contractions that are in the wall of our digestive tract, meaning the stomach, small intestine, large intestine. So those are a few examples of along the digestive tract. Then we're going to come back to the digestive system and talk about that. But those smooth muscle cells within the gastrointestinal tract, GI tract, rely on these gap junctions, allowing communication between neighboring smooth muscle cells. Now, the advantage of electrical signals is obvious. The advantage is speed. They are extremely fast. That's an advantage. The disadvantage is, and this is important, this is a major flaw, is when you use electrical signaling through gap junctions, there's no directionality to the message. Right? Directionality, directionality, meaning the cell cannot control the direction of the signal. So that's why the black arrow here, you see the two heads. Cell 1 and cell 2 have no control over that. So that signal can move back and forth randomly, both here as well. So that's why the arrow here, you don't have any directionality. And as far as the heart is concerned, it's fine. You know, when you want to activate the heart contract as a whole. when you basically contract the line, the tubing of within the GI tract, that's fine as well. But when, for example, you want to control your skeletal muscle, let's say you want to move forward. And if you rely on this type of electrical signaling, you won't move forward most of the time. You can move to your right, you can move to your left, you can move backward, you can jump up, you can jump down. There's no way for you to control it. So yes, it's fast, but the lack of directionality is really bad under conditions where you really want to control your motor responses, right? So let's say the car is coming, you cross the street, the car is coming, and you want to get out of the way, right? So you want to run away from the car. And if you rely on electrical signaling, you might run toward the car, and then you have no control over it. So because of this, electrical signaling mechanism is a minority among the signaling mechanisms. So most of the signaling mechanisms rely on chemical signaling pathways. So rely on chemical messengers, rely on chemical signals as opposed to relying on electrical signals. Because chemical signaling does have directionality. It allows the cell to send out a signal, and the cell knows for sure the signal moves to the desired direction. So this is the chemical signaling, and this is indirect because this one cell needs to secrete a chemical acting as a messenger, and that messenger has to bind to a receptor on the target cell. So here, it takes two to tango. So in other words, it requires a chemical messenger, but it also requires a receptor, right? So chemical signaling requires two things. Requires the chemical messenger and requires a receptor. And the interaction between the chemical messenger, the receptor, is exquisitely specific. It's beautifully specific. And you want that specificity. The analogy here would be the lock and its key. Okay? Exquisitely specific. So high specificity between the messenger and its receptor. So this is why, for example, the chemical messenger is released, for example, through the bloodstream, and then that chemical messenger will move throughout the body, yet only specific locations would respond to that chemical messenger. The reason is only a subset of cells will have the receptor that corresponds, that recognizes that chemical messenger. Any cell that does not display the receptor then will not respond to that chemical messenger. This is how the body controls through chemical signaling. So it involves the message, but it also involves a sensor, a receptor, right? And the binding between the chemical messenger and the receptor is not permanent. That's also another beauty. It's reversible. And the binding is not through covalent bond. This is through weak interactions, like hydrogen bonding. So this is no permanent covalent bonding whatsoever. So that chemical messenger can bind and unbind, can bind and unbind. So the receptor can interact with multiple messengers per unit time. And once the chemical messenger has done its job, it needs to be, deleted. It needs to be neutralized. That's another issue with chemical messenger signaling mechanism is you need to recognize, you need to kill off the messenger literally. So if there's a new message coming, the target cell can know about it. If you allow an old messenger laying around, then there's no way for you, the cell, to know, okay, this is an old signal versus a new signal, right? So all new incoming signals are by default new because any old messenger would have been neutralized already. As soon as they done their job, they would be killed off, right? So that's an important mechanism for cells to be able to recognize between old messages, incoming messages, between an old messages are already there versus a newly incoming message. So what you can say, this chemical messenger, you can say a chemical messenger, or you can say ligand or ligand. To this day, I'm not sure about the correct pronunciation. I've heard my mentors, some of them say ligands, others say ligands. So I have no clue the proper way to do this. But what is important is when you write, you need to spell it correctly. So it's Ligands, plural, and without the S in singular. So you can say chemical messenger, or you can say ligand. And the chemical messenger or ligand changes its name depending on the organ system that uses it, right? And so, for example, the chemical messenger used by the nervous system, it's called a neurotransmitter, shortened as NT, right? Neurotransmitter, one word, Neuro, transmitter, Transmitters. So two or more neurotransmitters with an S at the end. One neurotransmitter, get rid of the S at the end. So that's for the nervous system, neurotransmitter. If it's the endocrine system, then the chemical messenger is called a hormone, HORMONE, one hormones, two or more hormones, add an S at the end. And if the chemical messenger is used by the immune system, then it's called a cytokine. 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 One cytokine, two or more cytokines, add an S at the end for plural. Okay. So depending again on the organ system, cytokines for the immune system, hormones for endocrine system, and neurotransmitter for the nervous system. So these are the three main organ systems relying heavily on chemical messengers to communicate with other cells or with other organ systems. And we're going to learn later, when we talk about a chemical messenger here, we need to know whether that chemical messenger is water soluble, so WS for water soluble, or that chemical messenger is fat soluble, FS, right? So WS, water soluble, FS, fat soluble. If the chemical messenger is water soluble, then it will not be allowed to cross the cell membrane because the cell membrane is fat soluble, and water soluble chemical messengers are not compatible, will not be allowed to cross that membrane. So they would need a receptor that would be located at the membrane, the cell membrane as shown here. right? So this receptor is actually a transmembrane protein. So this transmembrane protein is located at the cell membrane, is located at the plasma membrane, where it will recognize a water-soluble chemical messenger. If the chemical messenger is fat-soluble, then that chemical messenger is allowed to get straight through the cell membrane, and the receptor would be located inside the cell, right? So that receptor does not have to be at the cell membrane. So this is for fat soluble chemical messenger, goes straight through the cell membrane, and the receptor is in the cytosol, or the receptor is in the nucleoplasm. So the receptor can be in the cytosol or inside the nucleus for fat soluble chemical messenger. But for the water soluble chemical messenger, the receptor is required to be located at the cell/plasma membrane. Now, chemical messengers for a short distance, for example, like the cells within microscopic distance from one another, that chemical messenger is called a paracrine signal. So example, paracrine signal growth factors, clotting factors, cytokines are used as short distance chemical messenger. So just simple diffusion, they move across, driven by concentration gradient. They leave the secretory cell and they attach themselves to the receptor on the target cell. There's another chemical messenger type called autocrine. Autocrine means it's the same messenger here, but instead of targeting the target, it goes back and it affects the cell that releases it. So it can induce its own secretion, or it can shut down its own secretion, right? In that case, we call this signal autocrine. Okay. So if the chemical messenger goes back and it can affect, it can influence the cell that produce and secretes it, then it's called an autocrine or autocrine signaling mechanism. Both paracrine and autocrine are short distance chemical signaling mechanism. And remember, I mentioned earlier, the cytokine or chemical messengers used by the immune system. So the immune system does not rely only on short distance. So that's why I wrote down here, not all cytokines function as paracrine. Some cytokines are indeed working as a long distance chemical messenger. So that's the concept of paracrine versus autocrine. So this is a chemical messenger used by the nervous system, so neurotransmitters. Throughout the semester, I'm not going to spell out the lengthy word here, a very long word. So most oftentimes, I'm going to shorten neurotransmitter as N, T, and S for plural, right? Neurotransmitters. The S is for this S here. So 2 or more neurotransmitters. And they're released by neurons. Now, the concept of presynaptic neuron and post synaptic neuron means there's a microscopic space called a synapse between two neurons. So you have this is a simplified version of a neuron. So you have two neurons. And there's a microscopic space here and so there's no contact between the two neurons. So this microscopic space is called a synapse. Right. So the blue cell, oops. The blue cell it's a presynaptic neuron. And the red cell, you have guessed it, is the postsynaptic neuron. Right. Pre because it's located before the synapse. Post is because it's located after the synapse. The synapse is the black grid here, reflecting a microscopic space. So there's no physical contact between the pre and post synaptic neuron. So literally, in that microscopic space, you would have chemical messengers released into it, right? So I'm going to do green here. So the chemical messengers are released. So each green dot is a neurotransmitter. Okay. So that's signaling, chemical signaling between neurons. So microscopic space between two neurons are called synapse. What about neuroglandular and neuromuscular junction? So let's say instead of the postsynaptic neuron, you don't have a neuron, right? You have a gland. Like a sweat gland, right? So this is multicellular sweat gland. So in that case, it's still a microscopic space. But that microscopic space is no longer called a synapse, and that's important. You still have that microscopic space, and now that microscopic space It's called a neuroglandular junction. Even though a lot of medical textbooks, use them loosely, all of it is synapse, but that's wrong completely. Synapse is only between two neurons, right? So if there's a neuron and an effector target, then it's called a junction. In this case, if it's between a neuron and a gland, it's called a neuroglandular junction. But if instead of a gland and you have, for example, a skeletal muscle fiber, right? So here, S, K, M, skeletal muscle fiber sense or slash cell, right? Skeletal muscle fiber or skeletal muscle cell. Then you also have a microscopic space, and that space, again, is not a synapse. That space is called a neuromuscular junction. So that's the difference between a synapse, neuroglandular junction, and neuromuscular junction. It gets on my nerve when I read textbook and then they use those loosely without making a distinction. For example, here they say target cell postsynaptic. In this case, this has to be a neuron. So this is not a secretory cell. This is a presynaptic neuron. So this here, this has to be a presynaptic neuron. And this here, it says target cell postsynaptic. Then this has to be a postsynaptic neuron, since we're dealing with a synapse, right? Synapse is between two neurons. So you have presynaptic, postsynaptic neurons. That's a synapse. So to be clear again, if you have a presynaptic neuron and a gland, then it's neuroglandular junction. Okay. If you have interaction between the presynaptic neuron and a skeletal muscle, then we have neuromuscular junction. In all three cases, the chemical messenger is still a neurotransmitter. Right, so in all three cases, the chemical messenger is always a neurotransmitter. So neurotransmitters are released into a synapse, neurotransmitter are released into a neuroglandular junction, neurotransmitter are released into neuromuscular junction. So neurotransmitters are always released by a neuron. What changes the cell after on the other side of the microscopic space, right? If on the other side, the interacting cell is a neuron, we call that microscopic space a synapse. If it's a muscle, we call it microscopic space neuromuscular junction. If it's a gland, we call that microscopic space a neuroglandular junction. So I just want to be clear here. For this applies for the entire remaining semester, because these concepts come back. Okay, make sure that you have this down once and for all. So erase everything that you've learned from anatomy. You most likely taught just is loosely called synapse. That's completely inappropriate, wrong. Okay, learn it the proper way. hormones. So chemical messengers used by the endocrine system, and hormones are for long distance communication. So when we talk about hormones, we're talking about released by cells of the endocrine system that we call endocrine cells. So this is for long distance. In other words, the chemical messenger is released into our vascular bed, blood vessel here, vascular bed. So when we say vascular bed, this is a term used a lot in the clinical setting. Meaning of blood vessels. So for example here, the secretory cell can be from your anterior pituitary gland at the base of your brain, right? I hope you remember where the anterior pituitary gland is. And it releases a hormone using the bloodstream, and the target cell here can be, for example, the adrenal cortex, right? So part of the adrenal gland that sits superiorly to the kidney. So meaning the gland that sits on top of the kidney, right? So the adrenal cortex is the superficial layer of tissue within the adrenal gland. And one particular hormone coming from the anterior pituitary can specifically target the adrenal cortex. We're going to talk about these hormones when we talk about the endocrine system, specifically steroid hormones, inducing the adrenal cortex to release steroid hormones back into the bloodstream. So hormones are specific cases where chemical messengers have to travel long distance before they get to the target. Same thing. In order for the cell to respond, it needs to have a receptor, right? It takes two to tango here. So if the target cell doesn't have the receptor, then the cell will not respond. So this here, that receptor is required. So cells with a receptor will respond to the chemical messenger. This here, non-target cell, it doesn't have the receptor, so it remains oblivious to that chemical messenger, right? So no receptor, no response. Even though we have the chemical messenger, the target cell doesn't have a receptor, no response. Okay, so those are examples. We're going to come back all of them. So insulin or ADH, antidiuretic hormone. So neurohormone mean this ADH is released by the hypothalamus and is stored by the posterior pituitary. So the posterior pituitary does not produce ADH. It just stores ADH on behalf of the hypothalamus. And since the hypothalamus is a neural structure producing chemical messengers that functions as a hormone, that's why it's called neurohormone. Whereas insulin is a bona fide hormone. Insulin is a chemical messenger produced by the pancreas. 99% of pancreatic cells have digestive functions. Only 1% of pancreatic cells have endocrine functions. And those endocrine functions are so critical, right? The beta pancreatic cells produce and releases insulin. The alpha pancreatic cells produce and releases glucagon. And we're going to talk about them later in the semester as to their importance. But briefly, insulin is involved in maintaining the concentration of glucose in our blood plasma. So let's say right after a meal, your blood glucose spikes, goes to the roof. Your cells cannot absorb glucose. It requires insulin. So your systemic body cells will absorb insulin, transporting insulin inside of them through the receptor-mediated endocytosis. This is transporting a VIP inside the cell. Insulin then induces the cell, the body cell, the systemic body cells to absorb glucose. So without insulin, our systemic cells, systemic body cells cannot bring in, cannot take up, cannot pick up glucose. In the case of ADH, vasopressin, or synonymously ADH, antidiuretic hormone, when you are being compromised in terms of fluid intake, when you are dehydrated, ADH is released, inducing your kidneys to basically conserve water. So basically you pee much less. This is, you see it when your pee turns yellow. So this is the body's way of basically conserving water under dehydration conditions. And we're going to learn the molecular mechanism specifically toward the end of the semester when we deal with the urinary system as to how specifically ADH can induce the kidneys to do this, can induce the kidney to reabsorb, reclaim water, to retain that water that's being compromised due to its intake. So we're going to learn about that when we get there. And these tables are important. It allows you to appreciate several of them. There are 6 tables. Table 5.1, 5.2, 5.3, all the way to 5.6. I strongly suggest that you learn them because they are fair game, even though they're not on the slide. So I'm specifically tell you during this recording that even though there's only table 5.1 and 5.2 on your lecture outlines, but you need to learn table 5.3 because there's a question to the end of this lecture outline. The answer is on 5.3. And then 5.4, 5.5, and 5.6, all these tables contain very important information describing the properties, the chemical properties of these chemical messengers. So this table, learn them as is. What I just want to talk about here is when we say lipophobic, it means hydrophilic. So it means water soluble. And when it says lipophilic, it means fat soluble. And we mentioned about this, right? So if these are chemical messengers here, these are different classes of chemical messengers. So if the chemical messengers are water soluble, they cannot cross the membrane. So you see the location of the receptor. They have to be on the plasma membrane for water soluble chemical messengers. But if we have the chemical messengers that are fat soluble, the steroids and the eicosanoids, They go straight through the membrane. So the receptor is in the cytosol or the nucleoplasm, meaning inside the nucleus. So we have the cytoplasm inside the cell, the nucleoplasm inside the nucleus, within the nucleus. So nucleoplasm, nucleoplasm. Right? So all fat-soluble chemical messengers go straight through the cell membrane, the plasma membrane, and straight through the nuclear envelope to get to the nucleoplasm. So you have here specifically the names, the neurotransmitter that's used by the nervous system, hormones and paracrine used by the endocrine system, hormones, paracrine. Paracrine can also be used by the immune system, and long distance chemical messengers used by immune systems as well. Over here, it defines for you paracrine means short distance. Neurotransmitter is also short distance. Hormones, long distance. Blood here means they are released into the vascular bed. Okay, VBS, vascular beds. Okay. Chemical messengers can be simple chemicals or can be complicated, can be a macromolecule like a peptide, like a protein. So it's a large molecule. It's a macromolecule. So proteins being one of the 4 families of macromolecules. So peptides and proteins can be used as a chemical messenger. Now, what is the difference between a peptide and a protein? Depending on the school of thought, depending on the textbook. So some textbooks, this is a convention. Any sequence of amino acid containing between 1 to 99 amino acid it will be called a peptide. If the sequence of amino acids is 100 amino acid or more, you guessed it, will be called a protein. So this is the one that we are using, this convention, because other medical textbook will say anywhere from 1 to 29 is a peptide. If it's 30 or more amino acids, it's a protein. But as far as we are concerned in this class with the Stanfield textbook, I don't even know if the Stanfield textbook relies on which method, but I am directing you to learn the peptide is up to 99 amino acids. From 100 or more of amino acids in the primary sequence, we will call it a protein. And when we say protein, it means the same as saying polypeptide. Okay, so protein or polypeptide are synonymous. So let's go over this. This applies concepts that we already learned about gene expression. So the peptide, when they are produced as inactive form, right? So that's why you see different names here. There's a pro-hormone, and then becoming the pro-hormone, and ultimately the functional hormone. So this is the functional hormone right here. This is functional. Okay. So there are specific enzymes, protease, that can cut off some amino acids along the chemical messenger. So pre-pro-hormone will have a sequence of amino acid being hydrolyzed, cut off. So you see here, it says cleaved amino acids, completely cut off. Cleaved amino acids, completely cut off to release the functional hormone. Okay? So an example here is the specific case, the parathyroid hormone. We're going to come back to this hormone. And the parathyroid hormone is involved in a very important hormone involved in maintaining calcium homeostasis. Calcium is the divalent cation, Ca2+, right? And the parathyroid hormone is produced by the parathyroid gland. And this gland is located posteriorly to the thyroid gland. So behind the thyroid gland, you have anywhere from 4 to 8 parathyroid glands per human being. Most medical textbooks will say 4, but that's not correct. You can have 4, your neighbor can have 5, your friend can have 6, another person can have 7, yet another person can have up to 8 parathyroid glands. And they all involved in producing, parathyroid hormone. And this parathyroid hormone is known as PTH. It is responding release when calcium concentration in our blood drops. And we're going to come back and learn about that. So there are 3 ways for parathyroid hormone to bring that calcium back up to under homeostatic levels in our blood plasma. And this is an example where the hormone is produced as a precursor and ultimately cleaved to release as a functional parathyroid hormone. And since the hormone, a protein, is released, it has to be translated by ribosomes that are associated with the endoplasmic reticulum and shipped to the Golgi apparatus. And then the Golgi apparatus will basically export it out, sent to the vesicles, and the vesicle will be exported out of the cell through exocytosis. So the exocytosis is an example of an active transport mechanism, right? So that peptide will have a signal peptide, and that signal peptide will tell the Golgi apparatus, hey dude, I am meant for export. So the Golgi will know and will ship it into the vesicle, and that vesicle will go to the fuse to the cell membrane, and using the mechanism of transport, active transport called exocytosis, the parathyroid hormone would be released into the bloodstream. So this is how the parathyroid gland produces and synthesizes and then releases parathyroid hormone. Okay. So combining multiple lectures, unit 1 to unit 2. So the process of gene expression, in this case, producing a protein, and this protein has a function, and its function is a chemical messenger. And this chemical messenger being released into the bloodstream for long distance, we call that type of chemical messenger a hormone. And since the gland, the parathyroid gland, is a member of the endocrine system, we are talking about the endocrine system as an organ system controlling all other organ systems. Only 2 organ systems control the rest of the body, the nervous system and the endocrine system. And this is a specific case of an endocrine function, controlling the body to respond to a drop of calcium in the blood. It's a beautiful, gorgeous story here. Okay. Oops, I forgot to erase. Now, moving on to the actual signaling cascades. So here, lipophilic messenger, we are talking about a fat soluble chemical messenger, right? Fat soluble chemical messenger. FSCM, fat soluble chemical messenger. If it's fat soluble, it goes straight through the membrane. So it can have the receptor in the cytosol waiting for it, or it can go straight through the nuclear envelope here, and it will have a nuclear receptor waiting for it. And these are very dangerous chemical messenger. These are very powerful chemical messenger. Since they are fat soluble, they go through every single physical barrier at the cell surface, which is the cell membrane or the plasma membrane. The barrier, the double membrane architecture of the nucleus doesn't matter. They go straight through the nuclear envelope and they have access to the nucleoplasm. So they have the receptor, they can bind to the receptor, and together here, when they have the hormone receptor complex, they become extremely powerful. Because in this case here, they can act as a transcription factor, TF. Transcription, that's the process when the DNA produce the messenger RNA. We learned that, right? Transcription factor. Transcription, let me spell it out. Okay. Transcription factor. Okay. TRANSCRIPTION, transcription factor, FACTOR. And the transcription factor can increase transcription or can shut down transcription. And here's why it's so important. We have DNA, and that DNA can be used to produce another type of nucleic acid called messenger RNA. Right? So you have your DNA here, and you have your messenger RNA here. And the blue arrow, that's a step that we called transcription. I defined for you earlier, transcription, right? T. So spell it out, transcription. So this step, transcription here, the transcription factor can come in and affect this transcription step. It can shut it down or it can amplify it. And this is important because when you produce the messenger RNA, when the messenger RNA gets produced, it will lead to protein synthesis. Right, so the protein here gets produced. So it can shut down the production of a protein, or it can amplify the synthesis of a protein. So that's why it's such a powerful chemical messenger. It has complete access to the control center of a cell, right? It can go straight to the nucleus. So any chemical messenger that is fat soluble is potentially dangerous. It can shut down protein synthesis. And if it shuts down protein synthesis, the cell will die, right? So that's why it's very important to recognize that. And that some of the toxins, are fat soluble. That's why they're so dangerous. So these are toxic chemicals coming from, for example, snake bite, okay, or any toxins that you ingest, or any chemical. For example, all these example would be all these medications that you take, and it affects the nervous system. And if they are fat soluble, they are very powerful. They're very powerful stimulant. Okay, so in this case, if the messenger is fat soluble. So HRE, hormone response element, HRE in this case acts as a transcription factor. And the transcription factor recognizes a specific sequence on a DNA molecule, which is a gene, right? A gene is a specific DNA sequence that codes for a protein. Okay, we're going to learn that later in the semester. So when we talk about a gene, it's a specific sequence of nucleotides, DNA nucleotides, that contain information to code for a protein. Now, this is an important thing here. We have only around 20,000 genes, yet we have more than 100,000 different proteins in our cells. So what it means is one gene can generate multiple versions of a protein. And that's exactly what happens. And we get to learn that also when we get there. Or we already learned it, right? I think. Let me double check here. we learned it already. So, okay, I'm basically confused here because my pre-med class, they learned that later in the semester, but physio, you learned it already. So I don't have to repeat this. Okay. I sounded like a fool, guys. Sorry. Okay, now what if the chemical messenger is water soluble? Okay, so you see here, chemical messenger, this is water soluble, so they cannot go through the membrane. So what this is important, the water soluble messenger remains outside of the cell. They cannot go in. So they have to convey the message through a transmembrane protein. So this is a receptor. So a transmembrane protein, TMP, acting as a receptor. In this case, it's a channel. So when the messenger binds to this transmembrane protein, which is a channel, it opens its pore. It allows electrolyte to flux through it, right? Sodium is coming in. Potassium is leaving. So let me, how do I do this? This here, this green arrow that comes in, throughout the semester, we're going to call this as influx. INFLUX. But the arrow that goes out, this arrow here, let's go out, we call it efflux. Influx, INFLUX, efflux, EFFLUX. So this is an F here, right? Efflux, EFFLUX. So all of this is passive transport. So all of these fluxes is driven by their respective concentration gradient, right? So for influx, I should have. So sodium and chloride and calcium, they all influx. So they all move through this arrow that points inward, right? So this is out of the cell. This is inside the cell. So extra means outside the cell, and cytosol means inside the cell. So when the channel opens up, calcium gets in, sodium gets in, chloride gets in, all driven by their respective concentration gradient. But when we talk about potassium, is the opposite. So potassium is efflux. Potassium is leaving. There are important consequences of all this when we go to the nervous system and the cardiovascular system. So when the channel open, potassium is leaving the cell. So potassium is going out. Again, driven by potassium concentration gradient, right? So all these fluxes are passive transport driven by concentration gradient. And more specifically, passive transport with the help of a transmembrane protein, we call it facilitated diffusion. Okay. So same thing here. This is what happens. This is the detail. So we have a water soluble chemical messenger. So water soluble chemical messenger, WSCM, WSCM, water soluble chemical messenger. Again, the water soluble chemical messenger cannot, I repeat, cannot get inside the cell. So they remain physically outside of the target cell. All they can do is they can bind to a receptor, and this receptor happens to be an ion channel. So when they bind, the channel opens, they flux. In this case, calcium gets in. Calcium is an example of a second messenger. So this is the primary messenger, and calcium is acting as a second or secondary messenger. So calcium gets inside the cell, but not the messenger. The primary messenger remains outside the cell. So once calcium gets inside the cell, it can cause several signals. The cell can respond. It can change how the cell responds through electrical signaling. It can induce contraction if the cell is a muscle cell. It can induce secretion if the cell is part of a gland. Or for example, if it's a neuron, it can induce the neuron to release neurotransmitter. It can also activate this passage here. For example, leading to smooth muscle contraction, right? So this muscle here, this is for skeletal muscle contraction. Or cardiac muscle contraction. But calmodulin is for, we say here, smooth muscle contraction. We are talking specifically about smooth muscle contraction. You learn from anatomy there are 3 types of muscle. I hope you realize this. We're skeletal muscle type, cardiac muscle type, smooth muscle type. And now we connect them into functions. So they have different ways of contracting. So the smooth muscle contraction is slightly different compared to the skeletal muscle and the cardiac muscle contraction. So that's the concept right there that I mentioned about the second messenger. It's written here. Second messenger. Okay. Calcium is one of 5 different types of important second messenger. So at the end of the lecture, I will ask you that question, and I will also tell you specifically where to look for in your book. So there's no excuse not to find them. Again, here, the chemical messenger is also water-soluble chemical messenger. It remains on the outside, right? Water-soluble chemical messenger. Now the receptor is unique. The receptor is also an enzyme. So here, it's a transmembrane protein that has a dual function. It acts as a receptor, but it is also an enzyme. That's why you see here the word kinase. Kinase means the ability to add a phosphate group onto another protein. And the name tells it's function, tyrosine kinase. So this transmembrane protein can phosphorylate the specific amino acid called tyrosine on a protein. So any protein that has a tyrosine will have its tyrosine phosphorylated. So the phosphorylation signal is a very important fundamental signal. It's like you turning on a light switch. So when the cell wants to activate a protein, wants to activate an enzyme, or wants to activate a target, all it has to do is add a phosphate onto the target. We call that process phosphorylation, right? So the tyrosine kinase adds a phosphate onto the tyrosine on a protein of the target protein. Tyrosine is a specific amino acid, right? So the 20 amino acid, tyrosine is one of them. On every protein, you will have several tyrosine amino acids. And particularly, some targeted tyrosine will receive a phosphate. And that's done by this transmembrane protein. This capability of recognizing a signal, recognizing a chemical messenger, and be activated by it, and the kinase function kicks in and it phosphorylates the protein, and the cell will respond down here by inducing protein synthesis or affecting its metabolic pathways, right? So this is another example of a water-soluble chemical messenger. And specifically, insulin. This is how insulin can act on its target cells. In this case, insulin acts as a hormone. And then here, this is a major family of receptors. They are called G protein linked receptors. They don't have a pore. And the messenger again is a water soluble chemical messenger. Again, the chemical messenger remains outside. It binds to this receptor, and you have here, this is the G protein. Okay, so that's the G protein. And the G protein has 3 subunits. You have the alpha subunit, which we're going to focus for this semester. We're going to ignore the beta and the gamma. So beta and gamma, we're going to focus on the alpha subunit. So once the messenger, the water-soluble chemical messenger binds to the receptor, the transmembrane protein, this is again the transmembrane TMP, transmembrane protein. And this transmembrane protein is linked to the G protein. That's why it's called G protein linked receptor, right? So the receptor is linked to a G protein. So once the chemical messenger binds and activate the receptor will activate the G protein. Specifically, the alpha subunit of the G protein becomes activated. And that alpha subunit can go on to activate an ion channel. So it opens an ion channel, and then it can change the membrane potential of the neuron, for example. This concept will come back in the nervous system and also the cardiovascular system. Okay, so a chemical messenger can indirectly open an ion channel. In this case, the chemical messenger could have activated directly an ion channel that we saw earlier, or indirectly, it goes to a different route. This is a contorted route. It's a longer route, activating a G protein coupled receptor to indirectly activate opens an ion channel. The difference is this channel has been opened by a signal that's coming from the inside the cell and not by a signal coming from the outside of the cell. So this is one mechanism by which the G protein can activate a response. A second mechanism is through the same concept, G protein coupled receptor. Moving on to this panel here. This is one of the major signaling cascade in a lot of species. The reason is because the cyclic AMP is one of the most important second messenger. And that's why I asked you here, what are the 5 major second messengers? We learned about calcium already. Cyclic AMP is one of the most, if not the most important of them. And these 5 major second messengers, you can find them on table 5.3, page 141 of your book. Right, so go to table 5.3, learn the 5 second messengers. Okay, so we went over calcium, we went over cyclic AMP. There are 3 more, and they are on table 5.3, page 141. So again, the alpha subunit of the G protein activates another transmembrane protein, and this transmembrane protein is an enzyme. All of this will come back, guys, so make sure you learn this. So it's called the adenylate cyclase, and that adenylate cyclase is an enzyme that can convert ATP into a cyclic AMP. So lowercase c, uppercase M, uppercase A, uppercase M, uppercase P is pronounced cyclic AMP. And this cyclic AMP, acting like a second messenger, it activates a protein kinase. This is another enzyme. And this protein kinase, the name indicates its function. It can add phosphate groups onto another protein, right? So the protein kinase will phosphorylate a target protein. So that's what you see here. This is the substrate, and it adds the phosphate to the substrate. And now this protein becomes activated, and this protein goes onto the cascade, leading to inducing the cell to respond, right? So that's an example. And this here, you go to page 143 of your book, and the figure is 519, and this is an important figure. The response is amplified. So the concept is called signal amplification. So the second cyclic AMP can amplify the response. So you can start with only one single chemical messenger, but you can produce thousands, if not hundreds of thousands of these signaling cascades. So the cell can respond. So the message was amplified, hundreds of thousands, if not millions of times. And that picture shows you the importance of this signaling cascade, the G protein coupled receptors, the G protein linked receptors, but the word link here is also used sometimes, it replaced by the word coupled. So G protein coupled receptor, coupled, COUPLED, right? And that's an important picture. Make sure that you learn that well. Okay, so that would end the recording for today. And then let's meet again until the next recording. In the meantime, stay well, stay healthy, be happy, and then study, right? Make me proud. And I will see you next time. Until then, take care.